Well, hey, biology. So this is our last notes for the year. I don't know how to be cool. Anyways, this is about shaping evolutionary history. So I hope we're doing them at home. I hope you dive into them, listen well, write well. We're going to review in class this week before our test. So without any further ado, let's get started. So shaping evolutionary history. So here's our objectives. So we're going to explain some ways evolution can happen as well. So like genetic drift and random allele changes, right? And then speciation as well as patterns of evolution. So here's genetic drift, okay? Genetic drift is a random change or random changes in allele frequencies from one generation to the next. In small populations, it can change allele frequency. So for example, you have this entire population of butterflies right here. And then by small population, and then these die by chance, and so all of a sudden you only have one color butterfly left. There's a couple different ways genetic drift can happen, random changes. One's called a population bottleneck, and so it's an environmental event that results in only a few individuals. So if you look right here in this jar, you can see that red and yellow, if I were to just ask you to describe the frequencies of each being, you probably would say, well, it's probably even 50-50, right? But all of a sudden, some change in the environment led to dramatically different allele frequencies. And now that population, well, it's mostly red now, right? Well, that is population bottleneck. And so this is an example of these chickens were very clearly seen around America, but fewer than 50 in the 90s. Little genetic variation due to hunting and stuff. Why is little genetic variation? Well, that's genetic inbreeding, and that will lead to a rise in diseases and disorders due to no genetic variation and variability in the genes. Bottleneck is another type of genetic drift, which, I'm sorry, not another type of one. Sorry, I thought we were on the next one. That bottleneck is the same one. Another example, I should say, sorry, first mistake, let's see how many I make during the video, is northern elephant seals, okay? So in the 1800s, they were hunted to near extinction. There's no variation in 24 genes. And so, look at that face. And so, it's pretty crazy. You can kind of, decreasing biodiversity and leads to possibly disorders as well, would increase disorders, okay? So this is, population bottleneck. You have all 20% types of alleles, and then all of a sudden, blue was completely wiped out, right? And so that could lead to evolution. The next one is the one that I thought we were on. Sorry about that. And it's the founder effect. So you have a lot of different genetic variation here, yes? These are all different types of alleles. A lot of variation here. Not as many here, especially if they don't bring in new outsiders or marry or have kids with people outside the community. So this is called the founder effect. And so this is when a few individuals colonize a new area, and it's equivalent to a large population being reduced by a bottleneck. It's just the opposite. We're starting with a small one. And so a great example from science, and you can watch videos, y'all are like the kings and queens of finding relevant videos on stuff, but Tristan da Cunha, I think I said that right, in 1415 British colonists founded this settlement, okay? But one of the early colonists carried a recessive allele, hopefully you know, for retinitis pigmentosa. 240 descendants on the island in the late 1960s, 4 had retinitis pigmentosa. You're like, well, that's still low, but that's way higher than the population around the planet, especially in Britain. And so this is the founder effect. You can look up this island. It's pretty crazy. Like, it's super safe. And, I mean, they're, pretty amazing, right? Like, I know during COVID, like, they never got it. Like, they were super strict, I guess. Or I don't know if they got it. So I haven't looked it up yet. But it's just an example of that. And so, for example, Amish populations exhibit founder effects. Maple syrup urine disease, yes, it sounds interesting, but it's not good at all. Much higher prevalence in children of Amish, Mennonite, and Jewish descent. 10,000 members of the Fundamentalist Church of Latter-day Jesus Christ of Latter-day Christian Saints community, which practices both endogamy and polygamy. 75 to 80% of the community are blood relatives of just 2 men. That's a founder effect. And so, a question for you: the flood in the Bible is one of the main events that has created the world we know. Was it a bottleneck or founder? And go ahead and pause. I'll tell you the answer here, but hopefully you have it as a bottleneck effect. Now, natural selection can act on quantitative traits in 3 ways. Stabilizing, directional, and disruptive. Stabilizing is when it favors the averages. Directional, when natural selection favors like one of the extremes. And disruptive, when it favors one direction, and then disruptive is both directions. Hey, mistake number 2. All right, so it kind of looks like this. The mean is selected for, a direction is selected for, and the extremes are selected for. So let's give examples to help us understand. Stabilizing selection is also called purifying. It goes on, it selects for the average. So for example, here's the average. And you can see percent mortality is in the line, okay? Optimal birth weight is about roughly right here, give or take. And so you can see mortality increases when you're super small or actually larger. And so this is purifying selection. Directional selection is when it's either positive or positive for either more or less of something. And so, for example, let's say this warthog is eating some this plant, but the number of spines, which are like sharp, prickly things, increases. And the warthog, of course, avoids that plant. And so you would see that, well, those genes would pass on to next generation and evolution would occur. And so that's directional selection. Directional selection can be seen in longhorn cattle, right? The longer the horns, the more protective you can be of your baby calf. Disruptive selection, individuals at opposite extremes. All right, so this is when natural selection favors opposite extremes of quantitative traits. So this results in increased variation, can result in bimodal distribution, which means there's 2 ways. And so an example of this is bill sizes in the black-bellied seed cracker, Pyranestes austrinus, maybe got that right. And so you can see right here in the place that they live, there's not many seeds of medium size. There's a lot of small and a lot of large, and so you got advantages here, but in the middle you're at a disadvantage because there's not many seeds with that. Okay, natural selection also can occur with a nut is when there's a change in frequency of a trait based upon the ability to attract a mate, and that's sexual selection. So the most famous example of this is the peacock. And so, right, the peacock, struts the feathers, and it's very colorful and vibrant. It has the eyes on the feathers. You can see that. And then the female chooses the prettiest bird, or the bird that, dances the best or sings the best or whatever. And then usually it means like a good ability to compete in these ways leads to good gene hypothesis. But that is sexual selection, okay? Now, if I showed you these 2 birds, you probably would think they are the same species. But they're not. They're actually different species. And they have distinctly different songs and other behaviors, okay? So a species is when you can mate and produce viable, fertile offspring that can also mate and reproduce, okay? And so for evolution to be true, we have to have speciation occur. We have to have, I'm going to try to do this with a mouse, but we have to have some sort of common ancestor, right? And then we branch off to species B and C, okay? And this would be a common ancestor right there. But this is also a speciation. That this is the formation of new biological species, usually by the division of a single species into 2 or more genetically distinct ones. So, again, species and created kind are not synonymous. Again, this says 6,000 years ago, so this is a young Earth reading. The Bible does allow for old Earth readings based upon my research and what I've said. But you can see kinds, right? The Bible doesn't say species, it says kinds. And so you have to have some sort of isolation. In order for speciation to exist, where species no longer mate and can produce viable offspring, you have to be isolated. And so that is reproductive isolation. There has to be some sort of barrier that prevents species from mating. And of course, this takes hundreds of thousands of years. We're not talking like rapid speciation here. And so there's pre-zygotic barriers. Remember, a zygote is something that is a fertilized egg cell, okay? And so pre-zygotic barriers block fertilization, okay? And so there's different types of these. The first one is habitat. They don't really encounter each other. So, for example, these 2 fly species. Well, one lives on apples and one lives on blueberries. Different habitat. Another pre-zygotic barrier is temporal isolation, which species that breed at different times of the day. They wouldn't mate and mix their gametes. And so the geographic ranges of the western spotted skunk and the eastern spotted skunk overlap, but they mate in different times of the year. Behavioral isolation is the rituals are completely different. We have completely different rituals, okay? So The infamous example of this are the blue-footed boobies, inhabitants of the Galapagos, mate only after a courtship display where the male shows off his blue feet, like, hey, look at my kicks. You can watch some funny videos about that. It's pretty cool. Mechanical isolation is when morphological differences prevent successful mating, so the anatomy doesn't fit together correctly. Gametic isolation is the sperm of one species may not be able to fertilize the egg. So this happens a lot in the sea, right, with corals or sea urchins or anemones, which when they release their sex cells into the water, why doesn't everything just fertilize? Well, you're missing some sort of protein, missing some sort of ability to fuse together, and so that is gametic isolation. Post-zygotic barriers is prevent a hybrid zygote from developing. So this is when a zygote is formed and something is born, but it has the inability to be a fertile, viable adult. So reduced hybrid viability, which means the hybrid's development or survival is extremely lacking. So some salamander subspecies of this genus live in the same regions where they may occasionally hybridize, but this guy is not very fit. Okay, they're frail, weak, unable to survive very well. Reduced hybrid fertility. So these hybrids are strong, but they are sterile. Most famous example in science is the mule. Okay, strong, very strong, infertile though. Hybrid breakdown. Some first generation hybrids are fertile, but as time progresses, they're feeble or sterile. And so this can be seen in this cultivated rice. So species 1, species 2, generation 1, generation 2, generation 3, weak, very weak. And so there they all are lined up right there, the different ones. And then finally, so when you have those barriers, those isolations, then it goes to speciation. So speciation comes in two different ways. Allopatric speciation, which means different country. Okay, and so this is when they're divided by a physical barrier. And this is the most common type. Okay, and so you can see right here that the canyon, Grand Canyon, these squirrels became different species. They look very similar, but they became physically isolated. Sympatric speciation occurs without a physical barrier. So this is rare, but it can be common in plants due to polyploidy. Polyploidy means many copies of a chromosome. And so here is an example of polyploidy. So you have a diploid plant, and it should undergo meiosis, but there's a meiosis error. And so instead of having haploid gametes, you have diploid gametes. So instead of it just being one copy in, you have two in. And that can lead to 4 in, and that could possibly lead to a speciation event, maybe even rapidly, right? And like we said before, strawberries are octoploids during our DNA labs, so they have many polyploidies. You can see right here, you can have a 3 in, non-viable or infertile triploid plant, because this plant would likely be sterile because it cannot pair up properly during meiosis. So you would almost have to have 2, right, to be able to do meiosis correctly. But polyploidy, and it's all because of a meiosis error. Okay, so that is our last lecture. We'll talk about it later in class. Hope you're doing well when you watch this. God bless. We're almost there. Let's finish strong. We're almost there.